All right, great. Next we have up Leo Ostruszka from Akadeum, speaking on Intro to Akadeum's Next Generation Isolation Technology.
Ostruszka. I've been working on this technology for 5 years now for Akadeum, and I'd like to thank Metroflow for giving me the opportunity to present to all of you today, and like to thank all of you for tuning in. So, as most of us know, our day isn't really always about performing the experiment we want to perform. It's about making the sample ready to perform that experiment. So over the years, various techniques have been evolved and discovered for taking what I'll call a crude sample, something such as blood or tissue or a mouse spleen, and turning it into something much more useful, because generally you don't want the whole, you want a pieces part. You want a specific target population, say Treg cells, or you want to capture specific DNA or RNA from a sample, or possibly just protein from a cell, because that's what you're really interested in, that subcomponent of the whole sample. So for years, there have been various methodologies to do this, things such as chromatography, columns, magnetics, and even flow cytometry sorting to help give us this improved or better sample. In reviewing all these technologies, Akadeum said, you know, there's got to be another way. There's nothing necessarily wrong with any of these things, but we want to try something else that might work in other scenarios and be more applicable for certain types of material. So we came up with the Akadeum Microbubble. Now, when I say microbubble, don't think your kids running around in the summer blowing through a wand and getting the pretty little bubbles that we get. What our microbubbles are actual particles. They're hollow glass microspheres. Now, we functionalize these microspheres by various means to either coat them with, say, streptavidin, another protein, an antibody, oligos, or just different functional chemistry on the bubble itself. So there's a lot of utility we can impart to the surface. As I said, they're hollow glass, so they're just full of air. They're also relatively large. In our case, it's a polydispersed population of approximately 15 micron. So if you put that together, large, hollow, it makes it very light. So these particles have a measured density of about 0.6 grams per centimeter cubed. So what does that mean? It means it will float in a biological solution. So water, PBS, blood, what have you. Our particles added to any of those solutions, they will naturally rise because they're inherently buoyant. Down below, you'll see two images of our microbubbles. The left is just a package of them, and the white layer is actually the microbubbles where they've all risen to the top. On the right, you're looking at an SEM image of a microbubble that has bound to a human monocyte, and that gives you a gauge of the size. So they're actually relatively large in the realm of particles, which most of us are used to thinking about. Now, I said they float. Their density is 0.6, so in water, this isn't water, this is a pretty generic buffer here. They will rise on their own, as you will see. I shake the bottle, goes to a cloudy mixture, and within a matter of minutes, you'll see that the vial will clear and all of the bubbles will rise to the top of the solution. Now, I will point out this was time-lapse. We speeded this up about 8-fold, but what you will see is in a matter of minutes, all of the microbubbles will go from being a homogeneous dispersed mixture to a layer at the top of the sample. And as you notice, there's no intervention. That's just sitting on a plastic platform. Nothing had to happen to make that go. The microbubbles will always rise regardless of where they are or what they're in. Oops. Now, as most people don't think about buoyancy very often, in fact, it's probably your high school physics class where you last heard about this, I'll give you a quick refresher. Buoyancy is essentially Archimedes' principle, which formally states the buoyant force is equal to the weight of the fluid displaced by the object in the given fluid. What does that really mean? If an object can displace more fluid than it weighs, it'll float. The math is real simple. The force is equal to density of the fluid times volume of the displaced object times gravity. So the beauty of buoyancy and the microbubble is it only relies on gravity. Gravity's pretty much always there. We don't have anybody using this in outer space or anything like that as of yet, but any lab, anywhere, you're going to have this work. Now, how do you actually use these microparticles or microbubbles? There's two general schemes that we've created. The first is a pre-labeled or pre-functionalized microbubble. This is handy if you're trying to remove a single type target, because all you do is take the microbubbles, add to your sample, mix, then wait. And what you'll see here is on the right, anything that is bound will rise with the microbubble. Anything that is unbound will generally sink to the bottom. Now, you can have a more complex modality, and that's demonstrated down below, which in this case, your sample is pre-labeled with an antibody cocktail. In this case, a biotin cocktail. After labeling the cells, mixing with streptavidin microbubbles will result in the microbubbles binding to those biotin-elated cells. And again, they will rise, everything else will sink. So depending on your workflow and what you're trying to accomplish, there's two different methods of using these microbubbles in a general fashion. I'll give examples of both later in this talk. Now, we've been playing with this, or I've been playing with this technology for about 5 years. We've released it commercially, and we found this is applicable to a lot of different areas. So it's not just our general immunology research. This technology can be applied to many other fields and many other things. We've seen applications in the cell therapy realm. We've seen applications actually doing direct diagnostics using the microbubble combined with a patient sample. And finally, we've seen things in chemical or bioprocessing environments where the microbubble can be added to either impart a characteristic or separate something from a reactor. For the rest of this talk, I'm going to focus on the first area, which is general research in primarily immunology, because this is a flow group. That's what most of us are most familiar with. So when we talk about immunology research uses, what am I referring to? Cell separation. When I say cell separation, what does anybody think of? Magnetics, or MACs. Magnetic activated cell separation. So on the left here is probably something everybody here has used, either a Miltenyi magnet and column setup, or something like BioLegend or Stem Cell with their magnet onlys. And we're used to having to hunt through the lab and find the stuff, or make sure, oh wait, do I have the right size column? Is it for my sample? Will this work? In contrast, the Akadeum microbubble technology, which we've coined the term BACS for buoyancy activated cell sorting, requires only the microbubbles and a pipette. I'll point out we don't provide the pipette. We do expect that most people already have those. So you don't have to worry about any ancillary equipment to either locate, find, wait for somebody to be finished using. All you do, open the microbubbles, add to your sample, you're good to go. Now, why would you want to use buoyancy as opposed to magnetics? Because, I mean, let's be honest, magnetics has been around for quite some time. It's fairly well debugged. The system generally works. Now, that being said, there are deficiencies. First and foremost, buoyancy is great because you don't have to worry about your container. The force of gravity is equal everywhere. So I can be working in a 500 microliter tube. I can be working in a 96 well plate. I can be working in a 100 ml jug or even a 5-liter reactor. And buoyancy will always be there. The bubbles will always rise. To contrast that with magnetics, you're very limited on the size of the sample you can process because the magnets get very large, very expensive, and cumbersome to deal with. Next advantage, as I already pointed out, is that you don't need any special tools or equipment. You need the bubbles. That's pretty much it. So again, you're not worried about where's my magnet, where's my column. Now, more importantly than those reasons, I think research recently has revealed some issues with magnetics. First, last year there was a really nice paper published in Cytometry Part A documenting that certain cell types are inherently influenced by a magnetic field. Now, this is primarily RBCs and monocytes. Now, RBCs, pretty obvious. Heme is heavily present. Well, what does heme do? Heme binds iron. What happens when you put iron in a magnet? It moves. So you can move the red cells. Now, what about monocytes? Well, monocytes will scavenge red cells. So in many cases, if you have monocytes or RBCs in your magnetically treated sample, you might either get cells you want or lose cells that you want based on these inherent characteristics. Another issue that's been revealed about or concerned with magnetics is recently people have shown that certain cells are actually eating the iron particles. Because on average with a magnetic system, those particles are down in the 100 nanometer realm. That is very easy to be uptaken. As I showed you earlier, the Akadeum microbubble is much larger. It's on the order of 15 microns. So when a cell interacts with it, it's not going to try and ingest it. It's going to treat it as a flat surface, say as a tissue culture flask or a vessel wall. Its response will be much more natural than if it sees this small particle. So this potentially gives you a happier or more naive sample because your cells are interacting with the particles less in a microbubble sample than they would potentially in a magnetic sample. Now, the final two advantages really go together, and what we've come up with is a very simple workflow that requires almost no handling, no container switches, and allows for multiple samples to be processed simultaneously. Again, with magnetics, you either need multiple magnets or multiple columns, and you're going from one to the next, making sure that you're not getting out of sync. With gravity and microbubbles, bubbles just rise. You walk away, come back, hey, look, it's done. You don't have to worry. And as I said, we've created a very simple workflow, which minimizes the transfer of samples and doesn't require much of an attention to detail. It's built with slop, as I like to say. Now, what does a typical workflow look like? So this would be the typical workflow for a general immunology kit. So here you're going to harvest your cells. In this case, sack a mouse, take its spleen, process that the way you would normally, mix with an antibody cocktail, just like you would with magnetics. Then you mix with the microbubbles. The mixing is a 1-minute step. I'll demonstrate that to you in a bit. Then you spin the sample. What that does is pellets your cells while accelerating the rise. At that point, you remove the microbubbles and you're ready to use your cells. In this case, I'm demonstrating just taking your cells to culture. So very straightforward, and what you'll see is there's nothing on this slide that you don't already have, or there's nothing on this slide you haven't already done. So the learning curve is very short. This is a very easy technology to work with. Now I'm going to run a quick video that will walk you through the process. Here what we have is a red cell contaminated RBC or PBMC sample. We add some microbubbles. Here's a quick demonstration of the mixing. Simple trituration with a pipette. We do what we call 30 strokes of mixing. Then we go to the centrifuge. After that, you'll have what's shown on the right is you wind up with a bubble layer at the top and a cell pellet at the bottom. This is very easy in the case of negative selection to then just remove the bubble layer, remove the fluid, and you are left with your enriched cell pellet. Very straightforward, simple process. Now, as I said, 30 strokes, you don't have to be exactly 30. It's flexible. If you lose count, it's not the end of the world. If you do 20, it won't perform as well. If you go to 50, it won't care. It will be just as good as 30. So it's a very simple mixing process as shown here. Now, next, I'm going to show you some data from several of our users. These will be mainly negative selection experiments. And I'll point out now that the images you are going to see are unmodified. So these are exactly as they were sent to us by the users. All I've done is added some commentary and description about what was done. So it's their data, not ours, and we haven't changed it in any way. So first is some data from Northwestern University, the Scott Lab, where they performed a mouse T-cell isolation. Focus on the bottom two plots, where we're looking at the final CD3 expression for purity. And this lab compared our mouse T-cell kit to BioLegend's Mojo Sort kit. And what you can see is both kits produced 98% purity, so equivalent result. But more interesting for this lab, what made them very happy was they also measured viability of their sample. And they found for their samples in their workflow that they had about 10% more viable cells as measured by Zombie Aqua. So that was a reason for them to say, hey, this is what we want to use. Another example here is a mouse B cell isolation. This is from the Zickerman lab at UCSF. In addition to showing just purity here, what this lab demonstrates is a functional equivalence. So first I'll start with the purity. And what you can see is comparing the Miltenyi sample here on the left to the Akadeum on the right. Miltenyi was 84% pure after the isolation. Akadeum was 81. So very similar performance. But more importantly to this lab is if you look at the IgG to IgM ratio and display, there is no difference between the magnetically isolated cells and the microbubble isolated cells. So again, here showing that not only does our technology perform to do a separation as one would want, it also gives very comparable downstream results. So we're not doing anything untoward to the cells. Final example here is another mouse B cell isolation, and that's shown at the top, where starting with a spleen at about 66% B cell, that was enriched up to 98%. But when this lab did, and this is from Derek Jones at Penn, these B cells were then induced to become plasma cells by exposure to CpG and IL-5. What that does, it induces the plant, it starts the induction of cells to become plasma cells, and that's measured by CD138 expression. And even though this lab did not share the competitive data, what they stated to us is the resultant expression of CD138 was similar to what they've seen using a magnetic product. So they got good purity, which they were expecting, but additionally, using the microbubble technology, there are no differences to downstream performance. So the cells are the same as they would be had they been collected with magnetics or some other methodology. Now I'm going to switch gears here a little. Instead of talking about a cell isolation, which have been, cocktails removing multiple targets, focusing on a pre-conjugated microbubble going for a single target. And this is our human red blood cell depletion product. Now this workflow is very similar, but one discrete difference. You'll see there is no antibody step because these microbubbles are already pre-labeled for human CD235. So a sample is harvested from a donor, red cells lyse or phycol performed, then the sample mixed with the RBC microbubble, same mixing as before, spun, remove the bubbles, then go downstream. Here showing flow cytometry. Now this whole process from beginning to end takes about 10 minutes. Now, why might you want to do this? Well, I think anybody who's worked with human blood and done phycol has been here and done that. Of you come in, or it's a Monday morning and you haven't had a whole lot of coffee, and you run your phycol. And you kind of did a lousy job. It's forgivable. We've all been there. We all know it happens. And in this case, a lot of red cells were left over in the sample, about 46% as shown in the top plot here. So that's quite a bit of that sample. Now, following the 10-minute process I just showed you, that becomes what I call a clean sample. And there's actually less than 5% RBCs remaining. Now, other than cleaning up how it looks, which is shown here, going from pink to clear, why would you care? Why does this matter? Well, what a lot of us do is we take these samples and we actually go to sort, because we don't want the whole PBMC fraction. We want a subset. We want the B cells. We want the Tregs. We want something, not the whole apart. And we want as many of those as we can get. So that results in us having to do a fairly long sort. Now, this is a recreation of data that one of our users does on a regular basis. We did it much shorter than what he does. So what I did here was an experiment to acquire 250,000 CD19 positive B cells on an Astrios. Now, what I did was compare two sets of samples, one that was pre-treated with the Akadeum microbubble and one that wasn't. On the left, without pre-treatment, what you'll see is about 10 to 15 minutes to do the sort. On the right, with the cleanup, that went to 5 to 10 minutes. So if you extrapolate that out, that third reduction in time actually will take a 3-hour sort to a 2-hour sort. In the case of one of our users doing this on a regular basis, it's knocking his 4 to 5 hour sorts down to about 2.5 hours. So why is that important? It's important for two reasons. One, would you rather have your cells sitting in a tube at the end of the cytometer for 2.5 hours or 4 hours? We all know the answer is 2.5. The cells are never happy there. You want to get them back into a normal environment as soon as possible. The second reason you would care, well, most of us have been there and we've sat there for a 6-hour sort before, and it's a pretty long day. If I can cut that time down, then why not? You can do a 10-minute process to knock off an hour or more. Why wouldn't you? That saves you time, gives you healthier cells, gives you better science, potentially saves you money as well. Now, one thing people commonly ask is, what happens after exposure to the RBC microbubble? Is it any different from lysis buffer, control cells, what have you? So what was done here is Jurkat cells were mixed with human RBCs. An RBC depletion was done or performed. The cells were put back into culture, and growth and viability was measured over 6 days. And what you can see here is there's no detriment from using the RBC microbubble. Cell growth is similar across all 3 samples, and the final viability at day 6 is shown out here. They're all about 82%. So no harm, no foul. That short interaction with the cell is having no negative impacts to it. Now, up to this point, I've been comparing the Akadeum microbubble technology to magnetics, because that's commonly how a lot of people are doing isolations. Now, interestingly, you can combine microbubbles with magnetics, because in many cases, magnetics are just fine. I did point out some potential problems, but it is still a valid, very good technology. But what we found is we can actually make it better. Now, I'm going to demonstrate this doing a fairly common isolation, which is the isolation of human NK cells. Typically, this is done with CD56. And in this case, I'm showing a positive sort of CD56 of NK cells using CD56 microbeads. And on the left, you see the starting control population. In the center here, you'll see, yes, this is an enriched population. I'll give you the numbers on the next slide, don't worry. But what you see in addition to the NK cells here in teal is the NKTs. Generally, you don't want those because they behave in a much different way than a standard NK cell. So what if you combine 2 methodologies, microbubbles for targeting CD3 cells and magnetics for targeting specifically the CD56 cells? In following that workflow, what you actually see is we've removed the NK cells. Now, there's 2 ways you can go about doing this. One is you can do one process, then the other. So start with a microbubble treatment, follow up with a microbead treatment. Or you can actually streamline that process and combine certain steps, because you have 2 different modalities for capturing cells. If you want to do a magnetic process followed by a magnetic process, you can do that. It just generally takes longer and you take larger yield hits. What we saw here with our combined modality is that not only did we have improved purity, and what was done here, 3 separate donors, 3 separate days, we found on average the magnetic particle alone resulted in a 64% purity. Not terrible, not great, but is what it is. In those same experiments, the combined the combined process improved that purity by about 11%, winding up with about 76 on average. So we got a much higher purity of the final NK population. But what I think is just as important is we actually didn't lose as many NK cells. So in 2 of the donors, there's a marked less loss of NK cells. And the third, it was a wash. They're about the same. So in this combination treatment, not only did we get a more pure population, we got more of them. And what does everybody want when they do sample prep? You want more of what you've got, and you want it as clean as possible. So these types of interactions are possible and are being done today by combining our technology with magnetics. Now, with that, I'll summarize, and I think I've demonstrated to you today that the Akadeum microbubble technology can effectively capture and separate cells. It's a very simple, straightforward workflow. You don't need any additional equipment or training. I've shown not my data, not in-house data, but real-world user data showing comparable performance to magnetic platforms. Additionally, I've shown that our technology can be combined with magnetic systems for improved results. And finally, there are some definite unique advantages and potentials to this microbubble technology. We have not exploited everything this technology can do. We think there is a lot more out there, and we're looking to grow it, partner with people, and find out everywhere where microbubbles can make sample prep cleaner, easier, and faster than existing technologies. With that, I'd like to once again thank everybody for tuning in, and I'd be happy to answer any specific questions about our technology.
Perfect. Thank you so much, Leo. We really appreciate it. That was a wonderful talk. And the Q&A has been lighting up, so we have about 4 minutes for a couple of these questions. So Lydia Tesfa had asked, can you sort microbubble attached cells? So is there ability to go ahead and use a cell sorter after you've used these microbubbles? And then she followed up with, can you use this technology for viruses and bacteria?
I'll start with the second. Virus and bacteria, absolutely. We actually started in bacteria back in the day. So yeah, as long as the microbubble is appropriately functionalized for your viral or bacterial target, it can be used to capture. That works actually fairly well. With respect to sorting or flow cytometry of a cell bound to a microbubble, it can be done. There are some interesting caveats about doing it. And primarily, the bubbles tend to rise immediately as you begin your sort. So they'll rise in your fax tube while you initiate the sort. So I've been much more successful just doing short experiments of cytometry, or fax analysis, as opposed to doing a full-blown sort. But it will work. You just have to keep mixing the sample, which is easier said than done while it's already on the instrument, because the bubbles always actively rise.
Yeah. Okay, great. And I'd imagine too that there's, based on how many bubbles are bound, might be a sizing issue and choosing the appropriate nozzle as well.
Yes, sample concentration has a lot of impact to it, and yeah, your nozzle size depending on how many microbubbles, how large are the cells, how many cells are bound. So there's some little games you have to play to make it work, but you can run them. As I said, a long sort is very difficult though, because your sample will disperse before you can run the whole thing.
Okay, great. Then we have a question from Ira asking, can you safely remove the cells and the microbubbles from each other? If you're looking to do a positive sort for whatever you would use to isolate, and how would you remove them?
We are currently, all of our products are negative selection or depletion based. However, we are working on a methodology for release so that we can have a positive selection portfolio. I'm not at liberty to discuss how we do that because it is a proprietary technology, but we are looking to be working on that more and releasing it in the future. You can try some basic things to remove cells from bubbles. It will depend on the cell type and how it was attached. For example, if you're binding with strep-av biotin, that's much harder to break than some other capture mechanisms.
Okay, great. We have 1 more minute left, so we'll try and squeeze in 2 questions if possible. So Thomas Miller had asked, are the bubbles toxic if cultured with cells after selection? But you just said that most of them are negative. And then do you see a certain time period where they dissociate from the cells?
Second question first. The auto dissociation or the cells falling off is cell type dependent. So certain cells will fall off more readily than others. I won't go into details there. As for culturing cells attached to bubbles and then what happens, or cells post-release from bubbles with our technology that we're working on, it's no harm, no foul from what we've seen. We generally see good viability of the cells. Growth is normal as compared to controls, and we've not seen any errant or large changes in certain cytokine expression. I'm not at liberty to share that data at this point, but I can speak to it to that level.
Okay, great. Well, that's the end of our 30 minutes. There were a couple of open questions, so we'll reach back out to you about those, and we'll make sure that everyone has that available to them. So thank you so much for a wonderful talk. It was great.
Thanks for the opportunity.
Of course.